It's my pleasure to introduce our next speaker. Dr. Richard Gibbs is the Wofford Kane Chair in Molecular and Human Genetics at the Baylor College of Medicine. He's a member of the Institute of Medicine and a fellow of the Hagler Institute for Advanced Studies here at Texas A&M. He is the founder and director of the Human Genome Sequencing Center at Baylor College of Medicine, and that happened in the mid-1990s. And the Human Genome Sequencing Center is dedicated to the mission of advancing medical care through research and translation of genomics. The HGSC was one of 5 centers identified worldwide to conduct the Human Genome Sequencing Project. Subsequent to that, the group has collaborated to sequence many species. I was involved with Richard as we set up the bovine sequencing project. And I remember very clearly that meeting in Houston where we worked out the details. The group also provided the first, or very close to the first, genome sequence from a single individual. So the original Human Genome Sequencing Project used DNA from several individuals, and they were all pooled, and it was unclear what the structure was within any one of those genomes. So the Human Genome Sequencing Center did the sequencing of senior moment here. Yeah, Jim Watson. Should have remembered that. The other genome that was sequenced about the same time was that of Craig Venter. Now, the 2 sequencing projects came up with similar conclusions in terms of numbers of genes and the depth of sequencing with comparable, and the difference was that the Baylor group accomplished their sequence for pennies on the dollar compared to what Celera's sequence was. And this marked a dramatic improvement in accessibility of DNA sequencing, because now lots of people could afford to do DNA sequencing. More recently, Richard and his group, which is a large group of 200 or almost 200 individuals, have integrated genome sequencing as a diagnostic tool in human medicine. And they've made some remarkable advancements in this particular approach. And so it's my pleasure to introduce Richard to you and learn more about how genomics can advance the cause of human health. So, Richard.
Hey, thank you, Loren. Thank you, everybody. Tremendous pleasure to be here, and a real honor, not just to honor Jim, but also to be here with the other speakers. I want to start off by really telling you how my association with Jim grew and what the substance of that was. I've put here a little timeline that says what the activities of the Human Genome Sequencing Center have been over the last 30 years. Initially, we started really with the mission of getting the human genome done. Some model organisms came along the way and led us into comparative genomics. And we continued with that until about 2012, when really that program ramped down because we were focusing more on the human genomes. So our engagement with Jim and with the bovine project in particular was in that period. And you can see here there's a map of most of the organisms or most of the species that we were able to do during that period, of which the bovine was one. So how did we really engage? Jim and George Weinstock, who was the co-director with me at the centre at that time, he'd moved across from the University of Texas in Houston. They worked together, and between us all, you see, we wrote this white paper that went to the NIH, and Harrison referred to that yesterday. Loren, I assume you did some of the work on this white paper too. I perhaps did less than I care to admit. But no, it was a persuasive document, and it really pointed out that, among other things, the bovine project was ripe to do because of the state of the reagents and the readiness of the community. So that was a persuasive argument in the NIH circle, and got the cow up to where the dog was, at least in the eyes of the National Human Genome Research Institute. And these two press releases echo those sentiments, although if you're able to read some of the fine text, you'll see that there were some money issues involved that had to be resolved until things really came to fruition. And that really was a period when I really gained an appreciation of Jim's influence and his reach. And it was no more apparent to me, this is a list of all the people who had to be coordinated, and believe me, some of them were not that willing to step up with the funds. But yeah, I got this realisation when we had that meeting in Houston, and Jim asked Anne Armstrong to make some comments. She was the then regent, chair of the Board of Regents of A&M, and a very influential figure. Part of the money here was to come from the Texas state, other money from the feds. And it wasn't all forthcoming. But Anne made a long speech. And the first thing she said, though, is, because George Weinstock and I were sitting in the front row, she said, this project's not going where it needs to, and it's really Richard and George that are holding it up. I was like, oh, crikey. And she went on for quite a bit about this, and she said, you know, they're really not doing what they need to do, and they haven't really focused on this appropriately. And I was just shrinking in my chair and about to leave when she said, so don't worry, I'm going to fix it. I'm going to call Dick Cheney and George Bush and tell them they've got to put the money in. So she wasn't talking about us, she was talking about the country leadership. So that gave me an appreciation of the reach of the Texas A&M community in the politics of the country and the complexity of the group. Anyway, Harris told you all the story leading up to how we got the project to go, and the project itself became this chapter in sequencing at the Genome Centre when we all put our backs in and generated the data and generated the assembly and brought it together. And I wanted to think of some comments to tell you how I distinguish the bovine community from the other communities, because in the course of these projects, we got to know a lot of other different groups, and each community around each organism actually has its own particular characteristics. And I'd say there are two things that struck me about the bovine community. One is the sheer readiness of the community to exploit the genetic data that arose from the fundamental foundation of the genome sequence. And I think that's echoed in the many publications and companies and enterprises and activities that have gone on since then. And the second thing is, as far as I know, this is the only community that ate the donor organism sometime after the project was done. But then perhaps I wasn't involved in some of those other projects. And Harris wrote a nice perspective on this when we published it. So that's my engagement with Jim, was really focused around that. And apart from that, just enjoying his incredible friendship and his being a gateway for us in Houston to the A&M community. So I can't miss this opportunity to thank two other elements of the A&M community. The first one is the Hagler Institute, and I don't know if John's here, and Cliff, of course. What an amazing thing that institute is, and some of you other guys have enjoyed that. And the second thing is Donna Musney. So Donna, who's here, started working with her in about 1990, or more than 30 years ago, and she's been foundational, and we've got a joke, she's the one essential thing in the Human Genome Sequencing Centre. And for those of you who are skeptical about her role, look at her publication record. We get these emails that you get about you have been cited in ResearchGate as the X number of the best person in the community. And to the chagrin of some others, she's the top-ranked woman in the Houston Medical Center in terms of her publications and scientific and research career. So thanks. And she, of course, embodies the Aggie spirit of dedication and decency and hard work. Thanks, Don. Okay, so my comments scientifically, did you all a favour at lunchtime? I threw away a lot of slides, so I'm going to be concise. Loren asked me to talk about the status of genetic testing, but actually you've heard a lot of that today. So here I am. So when we did the Human Genome Project, of course, it was full of great promise, and then it would drive genetic testing because the fruits of it would be the understanding of the architecture of human disease across the board. And the Human Genome Project has delivered an enormous number of things, in my view, and changed the world. But it has not given us that clue that allows people to go in the clinic and get information about common diseases. And this is the big deficiency. Oh, and I can't resist putting this slide in to remind folks that there are actually 3 genome projects. There was a 2000 event that people often cite as the draft, 2003, where we did this amount of finishing, and we did about 10% along with these other groups, and a bunch was done at the Sanger Centre in WashU. And then more recently, the T2T project, okay? And depending on which project you were majorly part of, that's the one you think was the real genome project. Okay, so we're short of these predictors for common disease. And, you know, after the Genome Project and the HapMap project, we had the initial GWAS that came out of the Wellcome Trust Consortium with signals that said, here are, you know, they look like Leif's pictures, the Manhattan plots, but then no follow-up in terms of finding the actual causative alleles that you can then take into the doctor's office and say, hey, I've got this allele. A real paucity of that. 20 years later, when we've got, you know, millions, more than 10 million genomes sequenced and these large cohorts all put together, we're still in the same situation. There are very few examples of causative alleles for common disease that have come out of the these human studies. And this is why, you know, I ask Leif questions like, hey, tell us where's the missing genetic signal? I can't emphasize how bad it is. If you had a heart attack, there should be 40 to 50% of the risk that got you there that is ascertainable by genetic means. That's from family studies and twin studies. But by molecular means, there's only a few percent of that risk that can be assigned. So we have this huge gap in this missing genetic signal in human studies. So what do we do about that? Well, in our environment, we have really, in a way, just not focused on common disease. We're focused on rare disease as the gateway to ultimately understanding all common disease. And to do that, we've developed technologies that we call personal genomes technologies, and Loren referred to some of those. You know, we were involved with the Watson initial project, and then a number of individuals, Jim Lupsky here, we sequenced him and solved his Charcot-Marie tooth disease origin mystery. And some other examples of individuals where we're able to actually provide therapeutic, actually a solution to, treatment for them. And then as these methods evolve, we developed these exome capture methods. We were able to move into the clinic and start doing diagnostics en masse. So in terms of the topic that I promised Loren I'd talk about, this is kind of where we got to developing these, and this is the last slide I'll say on that, on the topic of inherited disease. We've used those methods now to solve many cases of Mendelian disease. Okay, there are a number of programs. There's current one is the Grigor program, which is systematically using collections of research data to look at families and find Mendelian disease cases. Between us and the rest of the world, if there's 20,000 genes, probably half of them can be assigned as Mendelian diseases. Probably half of those are solved, but the other half remain unsolved. We've got some way to go. This has been going great for many years, until about 2 years ago, the numbers dropped off. This is data I got from Jessica Chung in Seattle, exome sequencing boosted Mendelian discovery. It's dropped off because nobody really wants to pay for it right now out of the research budgets. Still, overall, it's gone well. Now, with this Mendelian data, apart from solving those individual Mendelian diseases, they've done many other attempts to build oligogenic models to solve common complex disease, looking at burden of carriers and so forth. But none of it has really risen to the state where we can say, aha, we've solved the problem of the genetic architecture of human disease and the missing heritability. So we ask ourselves, where is that missing heritability? And Leif, prompted you to talk about the extreme polygenicity models. Perhaps we haven't found all of the kinds of variants that really will explain disease, that many of them may be in these regions that are less tractable in less than perfect genomes. Maybe there are some sort of expansion repeats we just haven't seen, so on and so forth. Maybe there's epigenetic signals that are not realised, and of course, as Steve was pointing out, maybe it's in gene regulation networks and various gene interaction effects that are yet to be uncovered. But what I want to tell you about today in the few minutes I got is how we might want to consider somatic mutation as another possible source of some of the missing heritable signal. Now, that sounds like an oxymoron, right? How does the somatic variation contribute to heritable genetic risk? And of course, the answer is there has to be some predisposition or some other effect. And it has to be kind of subtle, because if it was simply mutators or something like that, we would have found it, okay? But let's explore it. So we know that somatic mutations are abundant, right? When you are born, you get, maybe 100 mutations that you inherit, and then after that, every cell division is at least a handful in each division, depending on which cell lineage you look at in yourself. Then you've accrued, hundreds to thousands and thousands of these somatic variants, and they're there in your tissue. Everything depends on your tissue cycling and other issues to do with it. The individual part of you you're looking at, but there's a lot of them. So, and we already know that some of these certainly can cause what we'd recognise as simple genetic disease. There are, I think about 6 or 7 years ago, there started to be some excitement about Proteus syndrome, and then also about Vexus, which are two conditions which are actually somatic only. By the way, somatic, not malignant, somatic benign disease. And then there are other examples, such as Timothy syndrome and this lymphoproliferative syndrome, where there's phenocopies that are caused by the somatic mutations, little different from the heritable form in the same locus. And if you go to OMIM, there's a couple of hundred entries there that say somatic mutations can cause this disease, but OMIM is not really catalogued quite in the way that we need to get to a full systematic understanding. So what we can say is that somatic mutations definitely can cause non-malignant disease, but the fact is we don't know quite what the extent is. So we need to burrow into that. To burrow into that, we need to do some molecular detective work, and we need to develop new methods, and we need to build catalogues around these kind of events. Now, it's actually quite a tough molecular problem. And if you think about it in terms of this frequency curve, we're not talking about simple signatures where pretty much any old sequencing method can take a homogeneous tissue extract and get a little sample from a number of different cells and establish a signature. So you're all familiar with cigarette smoking from lung or skin from UV exposure, you can see the signature. And we're not talking about very early mutations, which are at very high frequency pretty much anywhere you look in any tissue. We call this the embryonic mutations. We're talking about the ones in the middle that might be present in just a small dysplasic part of a tissue that then causes physiological damage that then leads to precipitate the adult disease. And so you've got to identify those tissues. So we're making an entree into this field in a more systematic way, and I want to quickly tell you about 4 vignettes that relate to this. The first one was on the heels of a cardiovascular risk, heritable risk project that we did called Heart Care with the Baylor Care population, where we sampled individuals walking into the ambulatory clinics with some kind of chest problem and looked at a gene panel to ask, could we find markers that might say they actually had some genetic contributors to their risk? And the brochure here is all about the sociology of getting those individuals to sign up. So this is published, but I'll just tell you the basic result that surprised us was that 10% of the individuals that we got in the door and assayed in this way actually had persistent Mendelian high penetrance allelic variants that they'd had carried through into their adult lives and were contributing to their adult disease. That's this fraction here. If you thought about all kinds of other genetic things, some of the lipid markers and pharmacogenomics, we say nearly 30% of the individuals got some kind of genetic information from this study. But to us, the exciting thing was 10% of them had Mendelian disease. If you go to the general population, it's less than 1%, right? It's around 1%. So this is a real enrichment by testing in the ambulatory clinic. Now, what we asked ourselves was, what about those individuals who didn't have a genetic signal? Was there anything else we could find in them that might be in the category of a somatic contributor? And a graduate student then, Varuna Chanda, did some statistical acrobatics by looking at the low allelic fraction. This is like the number of sequence read counts. Here's the heterozygotes, here's the homozygotes. She looked in here for putative somatic variants. And I'm giving you the sort of high-level summary of each of these vignettes. She found 9 genes for which there was a convincing potential pathogenic signal that may contribute to their cardiovascular risk. Now, these are not simple chip stories, clonal hematopoiesis of indeterminate potential. These are true potential later mutations that actually can underlie these arrhythmias or structural malformations that you could constitute the cause of their adult disease. This is ongoing, but it's a signal that maybe screening in this area still would be advantageous. Okay, spurred on by this, vignette number 2, Eric Venner in the group, who's been working on epilepsy and heritable forms of epilepsy, looking for genetic signals in the idiopathic collection that comes through the UT clinics, decided to try and mine some of the large cohorts that we now have available because of the large-scale sequencing programs. And so he's been looking in the All of Us collection, where the Freezy looked at has about 250,000 whole genome sequences and accompanying metadata to see if there was, because we've got the allelic fractions that you can mine to see if there's a signal there that might reflect a somatic mutation. Now, difference here, in the HeartCare study, it's deep, it's panels, it's hundreds of sequence reads deep. Here it's whole genome sequencing, it's only 30 deep, so it's a harder task. And the short summary of what he found is that he looked in about 100 of these genes that are in the epilepsy class, in the sensitive regions of the genes. He was not able to find a significant difference between the signal he got across the whole collection versus the set of individuals in the All of Us program who had epilepsy and seizure in their metadata. So no enrichments, but he did find nuggets of individuals who clearly have a somatic disease. That is, like this guy who's in middle-aged and has a seizure disorder and a mutation in a gene that if he inherited that would be as an autosomal recessive, he would certainly have a severe neuronal disease. So there's a couple of other examples he's fished out since then. So the summary from this vignette is that there is some individuals with low-level seizure variants, and if we did deeper sequencing and more extensive surveys, we'd find some more, and this is yet another sort of low-frequency screening opportunity. Okay, vignette number 3. So we have these Mendelian collections where we've been solving the Mendelian disease family at a time, and what about looking in some of the unsolved cases there to see if we could find more Mendelian cases? And this is a program that was led by Jim Lupsky and Jennifer Posey with me. There, looking in these unsolved cases, going pretty quickly here, sorry, now not using homemade statistical methods, but just going to the latest commercial software that has a high sensitivity option, and looking in some of these unsolved probands. Andy Rivera and Chris Krakowski have fished out a number of new diagnoses by finding a low allelic fraction in these individuals. Here are the examples there of 2 of them. You can see the low levels of genes here low levels of sequence reads that contain these mutations and reflect potentially somatic disease. The phenotype matches the marker. The marker is also involved in heritable disease. It's validatable by independent orthogonal methods. It looks like disease, it's disease causing. Now, we have the challenge, like every Mendelian study, of proving fully that this is the causative allele in this individual. So there's 3 vignettes that tell us we've got somatic signals, we've got disease, we've got something to work with here. Maybe it's not really changing the needle in terms of all of the genetic contributions to all of these disorders. So now I want to show you something we get from going and doing a deep dive into a single disease. And here I've got to thank the Hagler Institute, because it was the support from there that was enabled me to expand some of this work. So this is a genetic disease that we identified fairly soon after we developed the exome sequencing methods. My sister called me. She said her new granddaughter's having a developmental problem. And so, I said, well, let's sequence the family. We don't know, there's no other indication that that'll help, but it can't hurt. And that's Molly, my grandniece here. And indeed, we found a de novo truncating mutation in a gene for which we knew little about in our exome sequencing of the trio. Okay, so the gene is AHDC1, the HE hook DNA binding containing motif protein number 1 on chromosome 1, and it has a long single coding exon and 7 exons in the whole gene. It's a typical neurodevelopmental disorder, and it has typical symptoms. You can only really find it by the molecular studies. So what we're able to do with the Hagler Institute support was to build this registry where we're up to around 550 families worldwide now who have this disorder, and a couple hundred of them are now with their full clinical data in our registry, and we've been mining that data and looking, doing laboratory studies to find the function of the gene, and looking at different kinds of mutations, all the things you do following up these kind of Mendelian stories. But and there are missense and deletion mutations that are potentially involved. But for the purpose of what I want to tell you about somatic variation is that the truncation mutations are really the pathogenomic for the disease. If you've got the mutation, if you've got a frame shift in the middle of the gene, you've got the disease. If you've got the disease, more than likely that's the molecular mutation you've got. Typically then, what will happen is an individual with one of these generalised symptom sets will present for genetic testing, and they'll have those features that I list there, and they will, I've lost the slide. I had the typical slide here. So typically the individual will go in and you'll get sequence data from the locus, and 50% of the reads will be wild type and 50% will represent an allele that's truncating. That's the normal story. You say this is straightforward. This is a case of Gibbs syndrome with a mutation in this gene. So imagine our surprise where we got an unusual case where we found that 25% of the reads represented a frame shift in one of the alleles. A second allele around the same representation was present, and then 50% of the reads were wild type. So we had 3 types of sequence reads. It sounds a bit like what we heard earlier about these zygotic mutations spinning off into 2 different resolved forms. So we followed this up. We went back to the family, we sampled buccal samples and now hair samples, and we retested and got more accurate statistical measurement. We're able to phase the mutation on the paternal lineage, even though we didn't have the father. It turns out the mother's haplotype was a common haplotype, whereas the mutation was on a very rare haplotype, and so therefore was almost certainly on the paternal lineage. And so what we've got here is this kind of situation where there's some kind of damage potentially coming in on the paternal side here, some repair event going on in the zygote, and it's resolving differentially into the first 2 daughter cells. And one of the truncations is in one cell and one is in the other, so you have these 2 genotypes for the 2 cells. And in the DNA level, it looks like this. This is the same story, really. Here we have the two positions of the 2 mutations. They're adjacent to one another. Here you see TAGCC, and here you see TAGG-C. So the two adjacent bases are deleted. That's what's resolved into the daughter cells. Pretty interesting from a speculation about what the DNA repair event might be, whether this is some kind of cytosine-linked event. It's not a CPG, but a cytosine-linked event, whether there's actually some secondary structure here, quite possible in that sequence, whether there's a lot of attention now on these ribonucleotide insertion repairs resulting from pool imbalances that can happen. And as I was just thinking after the earlier talks, maybe there's some kind of differential methylation or some other event in the sperm that's going through. This was a normal conception, nothing to do with IVF. But what else is interesting about this is the actual balance of the two alleles in the daughter cells. So we see then in the mature blood, these two cell types are marked, and they started at the zygote. They're now 50-50 in the mature blood. What happens between blastomere formation and the hemopoietic stem cell and its immediate precursor is a bit of a black box in our developmental understanding of the developmental lineages. And so the fact that they're 50-50 at this stage is of interest as a marker for those cell lineages. Of course, the hemopoietic stem cells come from the endothelial cells in the blood lumen and also from the bone marrow. And then before that, of course, they come all the way back to an earlier choke point in the lineage. So how big is that choke point and what happens? Interesting speculation about what happens to individual blastomeres. So I had to go back and learn my embryology to be reminded that a blastomere is different to a blastula. Right? Blastomere is the first few cells that come right after zygosis, and then what happens to those individual blastomeres, I think, is something that we still have to resolve some mapping. And I had good conversations with Alexei Abiov in the Mayo Clinic, who's used some of these early mutations to look at the selection of blastomeres going into the germ that can maybe have an influence on what damaging variants go into germ versus what goes down into the somatic lineage. Pretty interesting stuff, powerful markers. So this is not the only double mosaic that's been reported in human disease, and these are the other ones I was able to find in the literature. And if you look down the left column, you can see the list of disorders they're associated with. And if you look under blood, that's the fraction of the two alleles, and it's a bit hard to read, so I made a little chart for each one. So where the green is 50%, that's the wild type allele, and the other two colours are the two variant alleles. And you see in the top in the Gibbs study, they're about equal. And in the neuropathy, third last column, they're also about equal. But in the others, there's potentially skewing for different reasons. Now, there's sort of a story in every row of this table. For the Cornelia de Lange one, there's actually parental trisomy at that marker. So there's more to say, but the general point here is that these double mosaics are pretty useful lineage markers in this context. So wouldn't it be great if there were many more of them that we could find? And here, of course, we've found them in the context of disease phenotypes, which is a pretty narrow selection set. So we hope that many of the double mutation sites that we look at might actually be sources of these kind of markers. Aniko Sabo's been trolling through our TRIO data, looking at new mutations and looking at events that are close to one another. And she finds that virtually all of them are, unfortunately, on the same strand in the sequence data. If they're on different strands, they could be in this class, but on the same strand, it is almost certain that they're an event that hasn't resolved differentially into daughter cells. So drew 0 there, but there's still some of these. So what to conclude from this is that these from these 4 vignettes, to conclude that mining all these things does find examples of somatic disease that we haven't really appreciated yet, and we can find many, many more of those, and these can justify future diagnostic screening. And also just showing you that these things are valuable cell lineage markers. Now, is this going to blossom out and solve our question of where's the missing heritability? A bit early to say, at the best, I would say. But if we wanted to do that, we're going to have to bring this to a higher level and essentially mine more data. How did we do that ideally? Well, first we'd have to get access to more tissues from more candidates, from normals to build a catalogue, and ultimately from disease states. And then we need to use our better knowledge of cell lineage and use new sequencing methods with higher accuracy that can get into this, where this red question mark is, this part of the frequency curve that's so hard to get into. And of course, we need the software and infrastructure to do that. Now, happily, there's a program started to do exactly that. It's called SMAT. It's a NIH Common Fund program that still exists. And we're in the second year now, and its ambitions are to do exactly this. It's a consortial project. There is 5 sites that are generating the genome data and another 8 sites that are developing these technologies. And we're all working together to first benchmark these materials and then to generate the real data. So the program's ambitions are to generate a catalogue from 150 donors post-mortem, 10 to 19 tissues per donor, and perform all of these methods to establish this baseline catalogue as a first step. The tissues, the donors are what we call the healthy deceased, no cancer, no infection, no chimeras, and from diverse ancestries and gender and adults. The tissues representing all germ layers, and also some accessible tissues, blood and buccal swabs, so we can ultimately go back and make some correlates with what we find across the tissue set. And now the status of the program, you see in the top line there, we've, as a group, as a community, we've got together and we've standardised methods and language and communication and all that good stuff. We've worked through cell models to look for sensitivity of methods. We started with tissue homogenates. We're just now generating donor tissue data. And if you see that fancy diagram there, if you go to their website, it's actually a dynamic thing. You can see which centre is generating which assay, which data set. So this is the ongoing, and this is the work, this is the status of it. And I took out a series of slides that show you some of the early sort of technical achievements. I'll just show you one, which is from our own Chuck Song and his student Yang Zhang, which is a modified version of duplex sequencing, which is one of the ultra accurate methods where you look at the Watson and Crick strands and make sure they correlate. If they don't, you've got an error. If they do, you've got a real event in the primary molecule. And this version uses transposons to get full genome coverage, fancy acrobatics with oligonucleotides to make sure that the recovery is most accurate. And if you apply it to cord blood samples, you get this profile down here. Error rates, accuracy is around 10 to the minus 9. So it's kind of ultra accurate sequencing. Now the debate right now, of course, is the problem is not just the accuracy of the primary sequencing, it's the sampling you can do in the tissue locale. So it's still a technically unsolved problem, but we're accruing these data to get there. And then this will be a platform to extend into disease exploration. So my question for you, of course, is this is all in the human. So why don't you do a SMAT, like an animal version of SMAT? And then, of course, when I hear the talks I heard today, I realise you're kind of already doing a lot of that, but not in a systematic way, right? Now, don't believe that there's a program to build organ by organ in standardised groups of donors in animal genetics, animal genomics, but maybe it's a place to go next. So that's my suggestion. So to conclude all my comments, the Mendelian program's coming along. We should finish the Mendelian catalogue. Didn't say that so far, but I think that's absolutely something the community should do. But in addition to that, it's a golden era now for studying the benign somatic mosaicism, cell mosaicism contribution to the benign disease. And that's ongoing, and that's a great wave to catch. Whether or not this will end up being a part of the solving the mystery of the missing heritability, I think is probably a good thing to discuss. It's got to be a very subtle mechanism like the epigenetic state or something about the non-supergene haplotype. What do we call it if it's not the supergene? It's the opposite of that haplotype that might predispose you to being, the one who takes the second hit that really does have a somatic impact. It's got to be something subtle like that, but let's wait and see because the data will come. Anyway, thanks again for the invite, and I appreciate you all listening.
Questions?
start. So I've thought more about your question about the missing heritability. I don't think it's so much missing heritability, but the problem to find causal mutations. I think it's basically due to a misconception in genetics. I think we have been misled. But so where have we been successful in finding causal variants? It's in experimental organisms where you're cherry picking mutations that you could recognize when you do screening. It's in rare disorders in humans. You have strong hits that gives you strong phenotype, and it's in Mendelian traits. So you have large effect mutations. But in nature, this is the rare things. So we have a lot of variants that has a minor effect on gene regulation, protein function, and so on. So I think it's much more haplotype effects that we observe. That is clearly what we see in our herring models, that it's haplotypes affecting phenotypes. We have some strong effect, that is the rhodopsin, which I took as, but that is the exception. So I think if you have haplotype effect, it's so much more difficult to nail down causal variants, because these variants, small effect variants, come in different haplotype combinations, and we make it difficult to replicate them in different pedigrees. So maybe we should think a bit different how we evaluate these loci.
Yeah,
I could tell you one funny story. So yesterday, Harris mentioned Neil Copeland and Nancy Jenkins, which was important in comparative mapping. And they visited me in Uppsala a couple of times. And one was when we had discovered that dominant white in pigs is not a single hit, but it's two mutations together. And we had dinner that evening. I was excited to tell them this story. They didn't believe me. They said it's much more likely that you have a single hit. You're probably missing the true causal mutation. So this is the way how what I think we have been misled to some extent, because this is what we have been looking at.
Yeah, I don't disagree with you, Leif, but I would state it a little differently.
Okay.
I would state that the epidemiological evidence that there's a genetic signal is robust.
Yeah.
Family studies, twin studies,
I
etc.
completely agree.
And now we acknowledge that it's more complicated than we thought, but it's not intractably complicated, and we should be able to solve the mechanisms, even if it's not a rhodopsin. We should be able to understand these mechanisms, and until we do, we haven't reached the journey of understanding these genetic contributions. And I think there's kind of a bit of denial about that simple
maybe
logic.
the mechanism is more important than the causal variant effect.
Yeah. I mean, the mechanism can be much more than a single allele that screws things up. Yeah.
Thanks. I saw that you mentioned single cell on one of your slides. I'm skeptical, but could you speak towards how you might see single cell sequencing influencing next steps?
In the somatic...
In general, yeah.
Yeah, in understanding the somatic journey, I think, there it's the ultimate digitization of the somatic signal. I mean, the alternative is to take groups of cells and sample them as a group, and then you really don't know the distribution. Once you get into an allele, variant frequency that's less than 50%, it's in there somewhere, it's very hard to know how to assign it. So the calibrating everything by single cell technology, very valuable step, not really doable at this stage, including single cell RNA-based methods. So great value there, too pricey. Now, single cell, in terms of understanding the general biology that's linked to Mendelian diseases and their mechanisms, minimal contribution so far. I share your view.
Any other questions?
Thank you.